Happy to start.
Yeah, sure. Um, hi, my name is Jem. I'm one of the fourth years at Somerville. And today I'm going to be giving you a lecture on neurodevelopment and principles of neuronal function. I understand that this isn't possibly the most popular topic in second year. Definitely not when I was in second year myself, but I think it's quite interesting and it can be quite simple if you just try and grasp the very core like parts of it, essentially. So yeah, my email address is on the bottom left if you want to contact me about any questions. If you have any questions, you can just put them in the chat. And we can get started. So this is kind of what we will cover today. So we'll largely focus on embryonic development and the development of the CNS. And we'll do some things at the end on principles of neuronal function. So we'll discuss some of the st methods of study of the nervous system throughout the lecture itself, the things that are relevant to neural development. And I will give you a summary of the structure and function of neurons and glia, an overview of neuronal disorders, and some things on neuronal repair and regeneration. As you can see on the bottom left there, I've kind of color coded things according to whether the things that I consider that are on the syllabus, things that are clinical and things that are experimental. So yeah, let's get started. So in terms of the principles of neurodevelopment, so neurodevelopment is a really, really complex process, and the central nervous system comprises over 100 billion neurons. And it's not just the number of neurons that makes it so complex, but really the connections that they form that allows for kind of like the sophisticated function, especially in humans. And this kind of development begins in the third week of gestation, and it continues past birth throughout the first 2 decades of life. Uh, so in terms of, have you guys ever heard of the saying that the brain only fully develops at age 25? It's kind of, I guess, reflected of that in terms of how long it goes on. And it's really a reflection of how sophisticated the brain is. This whole process kind of involves patterning, migration, and then the connection of neurons and supporting cells to generate these local and global circuits, which are then generally throughout the first 2 decades of life, modified for function. And it's important that we kind of understand neurodevelopment because defects in this developmental process can directly cause certain conditions and has been linked to other clinical diseases such as schizophrenia. So what we'll be doing in this lecture is we'll be dividing neurodevelopment into parts. I've divided it into 4 parts. The first is neurulation and patterning. The second is neurogenesis and gliogenesis, so the generation of neurons and glial cells. The third is neuronal migration, so how those neurons and glial cells get to the locations they need to be. And the fourth is connectivity and network formation. So how these cells form connections with one each other to form the circuits that we are still really exploring, like experimentally and clinically. I've included on the right there a picture of kind of like the time frame that these processes occur within, and you can kind of see that it begins from conception all the way to essentially adulthood. Right. So first of all, I'd like to just do a brief recap on gastrulation. So gastrulation happens in the third week of gestation. And the cells, then it's basically when 2 cell layers become 3 cell layers, and the cells around the primitive streak undergo epithelial mesenchymal transition in waves. The first wave forms endoderm and then mesoderm, and then the remaining cells at the top form the ectoderm. And the ectoderm is really what we're interested in today because the CNS is derived from this ectoderm. Another important structure for, I guess, neurulation is the notochord, which is a mesoderm derivative. And gastrulation is important because it involves kind of like the patterning and establishment of bodily axes. So we'll go on to neurulation, which is the induction of the neural plate from the ectoderm. And this happens around week 3 to 4, and it begins with the induction of a neural plate. This is driven by the primitive node, which is the organizer in humans. And the primitive node secretes BMP inhibitors, such as cordin or noggin, which kind of inhibit BMP signaling that would otherwise drive these cells towards an epithelial fate. So these cells that receive these inhibitor signals adopt a neural progenitor phenotype, which is characteristic of the neural plate. So these cells will become neuronal cells instead of epithelial cells. One important piece of evidence that I quite like to include is that experiments by Spearman and Mangold in 1924, which basically just really concisely shows that the organizer is important in inducing the formation of the neural plate. Right, so what they did is, as you can see in the diagram on the left, they essentially retrieved the organizer region from one embryo, transplanted it into another, either directly into the wall or placing it within the embryo itself next to the wall, and found that both of these things induced the formation of a second neural plate and a second neural axis within the ectoderm surrounding the transplanted cells, and that really, like, I guess, really famously shows the importance of the organizer in inducing neurulation. I put down another study there by Cocker et al. In 2005, which basically just shows functional redundancy of BMP inhibitors, and you can read about it if that's something that interests you. So, in terms of neurulation, after the formation of the neural plate, um, the neural tube has to be formed through essentially folding of this neural plate from a 2D structure into a 3D structure. So from a plate into a tube, like a piece of paper, right? And this folding is induced by a sonic hedgehog gradient from the notochord, which is, what we were talking about earlier when I said the notochord is important in neural development. So what happens here is that the neural plate develops hinge points via like selective proliferation within these hinge points and essentially invaginates. And the neural folds at the very top, where they've kind of like invaginated itself, eventually fuse together dorsally, and this leads to the formation of the neural tube by the end of week 4. And that's exemplified in the diagram on the right. So rostrocaudal and dorsoventral patterning, so top to bottom and back to front patterning, is essentially driven by morphogenomic gradients. So in terms of dorsoventral patterning, it's driven by opposing gradients of sonic hedgehog from the notochord, which is ventral, and BMP or Wnt from the dorsal ectoderm. And this determines kind of the identities of the ventral and dorsal neurons. So the ventral ones adopt kind of a motor phenotype, while the dorsal ones develop a sensory phenotype. And you can see that still in adults because we have, dorsal root ganglia, which are sensory, and then that connects to the ventral roots, which are motor neurons. And this was kind of shown by Erickson et al., which basically showed that Sonic Hedgehog was responsible for this kind of patterning and was a morphogen that was important in ventralization and dorsalization, right? That's something that you can read about again in your own time. I won't go too much into it, but it's quite an interesting one to add if you wanted to add some evidence into your essays. And rostrocaudal patterning is determined by a combinational code of Hox genes, which I think you probably will have covered something along those lines in first year. So neurulation is important because it also has clinical consequences. And two of the major clinical consequences are anencephaly and spina bifida. So if the neural tube fails to fuse at the top, and it starts to fuse from the middle and progresses both sides, so it progresses top and bottom. But if it fails to fuse at the top, it will cause anencephaly, which is essentially where fetuses lack a functional cerebrum and a major portion of the skull or scalp, and that normally leads to miscarriage or stillbirth. It's not survivable. A failure of caudal vision, on the other hand, is somewhat less is generally speaking less catastrophic, and it leads to spina bifida, and that has three types of severity basically, and you can see that on the left there. Spina bifida occulta, which is essentially where the vertebral processes at the back don't fuse, but the spinal cord and the meninges still remain, and that's normally largely asymptomatic, but there can be a tuft of hair on the back. Meningocele is where the meninges kind of like spill out. And then the most severe form, myelomeningocele, is where both the spinal nerves and the meninges are evaginating essentially from the spinal canal. These kinds, well, neurotract defects have been associated with lack of folic acid, essentially. And these defects have been shown to be preventable through supplementation with folic acid. And on the bottom right, on the bottom left there, I've included a snippet from the NICE guidelines, essentially saying that pregnant women are advised to take folic acid supplements to essentially prevent neural tube defects. This has also been fortified in the UK through things like flour, so dietarily. So from this neural tube forms vesicles. So at the head end, so at the cranial end, vesicles form. So these kind of like I guess little bulges form to form three primitive vesicles that later develop into five secondary vesicles that give rise to brain, cerebella, and the ventricular system. So you can see on the diagram at the bottom. So essentially, first it divides into the prosencephalon, the forebrain, the mesencephalon, which is the midbrain, and the rhombencephalon, which is the hindbrain. The mesencephalon becomes the midbrain essentially. But the other two divide into two other vesicles each. So the prosencephalon becomes the telencephalon and the diencephalon, and the rhombencephalon becomes the metencephalon and the myencephalon. So, and then it's not just that, these tubes stay as kind of like a vertical embryonic folding brings these different parts of the neural tube and, the eventually the adult structures that they form into the right kind of relation with each other. So here I have diagrams of the adult brain and what they kind of correspond to. So the telencephalon becomes essentially the cerebral cortex, the thalamus, and the hypothalamus. The mesencephalon becomes the midbrain, and the hindbrain, the metencephalon, and the myencephalon becomes the pons medulla and cerebellum. So, not sure how popular this will be, but I have created kind of like a small interactive MCQ for you, which is on slido.com. This is the code, and this is the QR code if you want to scan it. I'll just start the interaction if you guys want to participate. So the question is, which of the following structures arise from the diencephalon? The cerebral cortex, the midbrain, the thalamus, the medulla, or the cerebellum? And don't worry if, you don't know at all, but uh, it's just one of those things that if you memorize it, it'll be fine. So yeah, you're right. It is the thalamus. And this is just a summary table of the different parts and the primary vesicles, the secondary vesicles, and the adult derivative. So as you can see here, the diencephalon gives rise to the thalamus and the hypothalamus. So in terms of the next part, which is neurogenesis and gliogenesis, two important things here are radial glial cells and the ventricular zone. So I'll explain what they are. The neuroepithelium of the neural tube, right, is composed of a pseudostratified layer of neural progenitor cells. And when I say pseudostratified, I mean it's basically one layer of cells, but their nuclei are at different places at different heights. So it looks like it might be multiple layers of cells. And these neural progenitor cells are called radial glial cells, and they reside like luminally within a zone called the ventricular zone, right? And these radial glial cells have, when I say that they're in a pseudocytoid epithelium, an interesting feature is that they undergo interkinetic nuclear migration, in which, like, basically throughout the cell cycle, the nucleus of these cells will move from one end to the other, which I think is quite cool. So DNA replication occurs at the peel surface, or the outer surface, and division occurs at the ventricular surface, or the inner surface. And these radial glial cells act as progenitors of both neurons and glia at different stages of development. And I do have a piece of evidence which shows this quite nicely later on. So, these radial glial cells can undergo symmetric and asymmetric division. And this kind of difference is essentially that symmetric division produces two progenitor cells, while asymmetric division produces one progenitor to renew the progenitor pool and one differentiating neuron, right? And as the central nervous system develops, there's a gradual shift from symmetric to asymmetric division, because you need to amplify the progenitor pool before you essentially specialize. Um, the type of division has been shown to be predicted by the kind of like cleavage plane, due to asymmetrical distribution within a cell of the molecule Notch1. So essentially, as you can see, and this was shown by and this was shown by Chen and McConnell in 1995, and I've included kind of a diagram on the right to help you better visualize this. The pink part is Notch1 distribution essentially, and you can see that it's asymmetric, right? It's all at the bottom. And what Chen and McConnell did was they fluorescently labeled these radial glial cells and observed that essentially vertical divisions produced two cells that behaviorologically were the same and were morphologically the same, using kind of like various different markers against cellular cellular markers, whereas horizontal divisions produced two cells that differed in behavior and kind of like cell surface receptor expression. And this can essentially be explained by vertical vertical divisions distributing Notch1 equally between the two cells, whereas horizontal divisions, obviously Notch1 is only distributed in one because of its asymmetrical distribution. So one kind of two kind of key hypotheses here in terms of cortical development are the protomap and the radial unit hypotheses. So at the stage of development, we're thinking that radial glial cells divide and they give rise to, all the neurons and all the glia, right? And at this point, we're thinking, people have asked how, like, at what point do these various different neurons and these various different cells know where they're going and what they're going to become, right? And that's what the protomap map hypothesis addresses. So it specifies that different functional cortical areas later on are initially patterned through molecular gradients in the stage of development, essentially establishing a protomap of positional information that's inherited by daughter cells. In the 1990s, this was contested by the protocortex hypothesis, which essentially stated that kind of an undifferentiated cortex only undergoes kind of a specialization for their later functions through external inputs that develop much later on. But this was essentially kind of disproven. And one of the major factors in disproving this hypothesis in favor of the protomap hypothesis was the discovery of cortical gradients of transcription factors such as PAX6 and EMX2, which act as kind of a developmental template for like location and establishes the protomap. Another kind of, I guess, symbiotic hypothesis is the radial unit hypothesis, which essentially states that the cerebral cortex develops as an array of cortical columns that originate from the ventricular zone through a radial glial cell scaffold. So you can see on the right here kind of a summary of these two concepts in which the bottom is the progenitor cells and the top is the cells that they divide and become. And you can see that through the color, you can see that these are different colors that have been programmed to become essentially different functional areas. And each of these columns still corresponds to one essentially radial glial cell scaffold. And this will make a bit more sense when I start talking about migration. So we've talked about neurogenesis to some extent. Now we're going to talk about gliogenesis. So radial glial cells eventually undergo a transition from neurogenesis to gliogenesis, and they give rise to astrocytes and oligodendrocytes in about week 16. The mechanism underlying this transition is, I don't think you really need to know this, but it involves demethylization of glial cell-specific genes induced by neuron-derived signals. And that's a study that, if you're interested in it, you can look further into it. But essentially, it guarantees that this transition into gliogenesis won't occur until it gets neural signals, essentially showing that there are already neurons having been produced. Importantly to note, microglia, which are another type of supporting cell in the brain, are mesoderm derived, and they migrate to the brain via vasculature during development, so they're not produced by gliogenesis. So let's go on to talk about migration. So from one layer of cells, you have to essentially, from one layer of neural tube cells, you're kind of trying to derive many multiple layers. So, for example, the cerebral cortex ends up in the adult with 6 layers. So how do they get there, right? So one way in which they get there is radial migration. So that's when neurons move perpendicular to the surface of the brain, so they create the layers themselves. So consecutive waves of migration form 6 cortical layers in an inside first to outside last pattern. And this was shown in a study by Angevine and Sidman in 1961. They injected pregnant mice with labeled thymidine that will incorporate into the nuclei, with DNA synthesis at various points of gestation. And then they harvested tissue from mature mice, showing that later injection corresponded to more superficially labeled cells. So essentially, the first couple of layers of cells were deeper within the brain. And this process can be summarized in the diagram on the right. So essentially, you get all of these different types of zones, and each migration forms a new layer, if that makes sense. So defects in radial migration, quite frankly, there are many of them. It's a complex process. It can go wrong. There are a lot of genetic defects that can cause defects in cellular migration, and these can have really catastrophic consequences for development. So one interesting one is X-linked periventricular heterotopia. So a defect in leading edge extension because of a filamin 1 mutation causes essentially neurons to accumulate within the ventricular zone. And in adulthood, this essentially is reflected, or even after birth is reflected as neuronal nodules lining the ventricular surface because they weren't able to migrate further out. Lissencephaly is something that's listed in the syllabus. One of the causes of lissencephaly is a mutation in a microtubule protein called LIS1, and this basically disrupts late migration and causes a smooth brain lacking sulci and gyri. A reelin mutation, on the other hand, I remember the lecture went on and on about this when I was in second year. But a reelin mutation causes cortical inversion, and this was kind of shown by Cavaness et al., who also, similarly to the previous study, injected pregnant mice with thymidine, and they showed that in reelin mutant mice, the essentially the order in which these layers forms were inverted. And I've given you kind of like a summary table on the right there, because there are quite a lot of things. Another type of migration is tangential migration, and GABAergic neurons undergo this kind of tangential migration. allowing for dispersion from where they are initially within the ganglionic eminences to the rest of the brain. So as you can see on this on that diagram, the ganglionic eminences are where they initially are, and these cells migrate parallel to the surface of the brain to get to their end location. Precursor cells from different eminences supply different regions of the developing brain. Again, you don't need to know the specifics, but I have listed them there. And this is a process that doesn't really require radial glial cells. So, like with radial migration, where they kind of like latch, where each successive cell latches on to the radial glial cell scaffold and pulls itself up. It doesn't require that. But it similarly involves axonal guidance, extension of the leading process, nucleokinesis, and a stop signal, essentially. So let's go back to Slido. Um, and this question is, in what order are the cortical layers formed? So are they formed inside first, outside last, with each layer closer to the ventricular zone? Again, the same, but with each layer further from the ventricular zone, or outside first, inside last, with each layer closer, or outside first, inside last, with each layer further from the ventricular zone. I understand that that's a bit of a mouthful, but yeah, you're right. It is inside first, outside last, with each layer further from the ventricular zone. So each neuronal cell migrates progressively further from the inner surface of the brain, so the ventricular surface, basically. So we're coming on to the last part of neurodevelopment, where I start talking about connectivity and network formation, which I think is honestly quite fascinating. So once neurons reach their final destination, they begin to establish connections, right? They begin to establish synaptic networks, and this happens through the extension of growth cones. And the way that these growth cones are guided is that they are attracted by chemoattractants and repelled by chemorepellents. These molecules can interact. And it also depends on the phenotype of the neuron itself. And this was shown by Song et al., who found that essentially activation of a signaling pathway in a neuron could change the attraction of a neuron to a certain signal. And these initial connections that they form are often transient, imprecise, maybe non-functional. So it requires regulation through neuronal death, axonal retraction, synapse elimination, and this is really important for neural efficacy and specialization. And these kind of connections are pruned essentially, based off of neural activity. So initially, there's spontaneous pattern neural activity that occurs, which is later sensorily driven, which can alter areas of the protomap. And this can be seen in terms of like senses when, for example, with eye opening in infants. There's extensive apoptosis of transient structures within the brain, such as the subplate, which is essentially a layer of the brain which acts as a scaffold or a template, but it is ultimately not needed. So that undergoes apoptosis, and it occurs as necessary to refine developing circuits. And this is a process which continues long after birth, alongside myelination, as we saw earlier in the lecture, with peak cortical thickness reached around age 11 to 12. I'm a little bit biased because I did do a research project on schizophrenia. But schizophrenia has been hypothesized to occur due to disruptions in this process. And when I say this process, I mean the pruning of essentially unneeded or inefficient connections. So Sekar et al. In 2016 showed that mice lacking functional C4, which is a complement component, showed reduced synaptic pruning during postnatal development, compared to their wild type littermates, which suggests that C4 regulates neurodevelopment during adolescence. And this is really interesting, because C4 as a complement component has long been used to essentially link schizophrenia to immune involvement. But the idea that it can actually have an impact in neurodevelopment through cortical pruning, probably through, for example, through microglia, is something that has really, I guess, reinforced a neurodevelopmental model of schizophrenia. Schizophrenia patients in so this was in mice. This study was in mice, but in humans, schizophrenia patients have been shown to have reduced synaptic numbers, overactivity of complement 4, and hence increased pruning, which may explain this link in humans. So it provides some sort of translatability from mice to human models. And that's quite important because in a lot of neurodevelopment, it's unethical, and a lot of development in general to conduct experiments on human embryos. So we largely do it in animals and try and see what we can learn and apply to humans. So when does neurodevelopment end? So in adults, neurogenesis is restricted to two areas of the brain, which is the dentate gyrus of the hippocampus and the subventricular zone. And while brain connectivity is established through the process that I covered in the last slide, synaptic changes still occur, but these are through long-term potentiation and long-term depression. So essentially, it's synaptic plasticity. And this synaptic plasticity can be shown again in a study by Jenkins et al. In 1990, where he trained a monkey at a task that required repeated use of certain digits and found an increase in the corresponding area of the brain. So we've kind of reached the end of the developmental portion of this lecture. Do let me know if there are any questions. Just pop them in chat or anything. Um, but for now, I'll go on to just giving you a summary of neuroglia. So three types of neuroglia: astrocytes, oligodendrocytes, and microglia. So astrocytes are these star-shaped kind of glial cells, and they have a lot of different functions. They essentially support neurons. All of them support neurons. But astrocytes do maintain extracellular ion homeostasis. They uptake and they recycle neurotransmitters. They promote synaptic plasticity. They control blood flow. They maintain the blood-brain barrier. And importantly, later they inhibit central nervous system repair and facilitate the formation of glial scars. And astrocytes are something that come up quite a lot in various different pathologies, including neurodegenerative disorders and yeah, I mean various different pathologies. Oligodendrocytes are the cells that form the myelin sheaths around neurons. Their main function is myelination, but they also support myelinated axons. And you can see that in the middle here. Microglia are small cells that are, I think, immune derived with processes that survey the surrounding environment. So they're the immune cells of the central nervous system, and they're responsible for things like phagocytosis, cytokine production, synaptic pruning, like we said in schizophrenia, and activation during injury or disease to mediate either repair or inflammation. Yeah, so that's a summary of all the neuroglia that you need to know about. In terms of regeneration and plasticity in this central nervous system and the peripheral nervous system, the main difference is that the ability of the central nervous system to repair itself is really inhibited because of an inhibitory environment. Whereas the peripheral nervous system has kind of a supportive environment for external regeneration with various different growth promoting factors. So as a consequence of that, the central nervous system has to essentially require to rely on plasticity to achieve functional recovery, whereas the peripheral nervous system has the ability to regenerate, and as long as they can reform that connection, you will get functional recovery. In terms of plasticity, the central nervous system is a bit more plastic because it's a bit more complicated. So there are a few more, you can imagine that there are more workarounds in a more complicated system, right? You can go through different pathways. And this is particularly evident in developmental and critical periods. So children often, recover from brain injuries better than adults if there's been a functional impairment. So in terms of functional recovery, the central nervous system is just dependent on plasticity and the ability to adapt intact circuits for this function, whereas the peripheral nervous system you can achieve re-innervation of targets, and it also has some synaptic plasticity, although not as much as the central nervous system. I hope that's clear and brief. In terms of neuronal disorders, there are two types of things that the syllabus wants you to know about. One of them is Rayleigh degeneration, which occurs after axonal damage. So axonal damage, when it causes disruption to axonal transport, leads to essentially Rayleigh degeneration, and the breaking down, which is the breaking down of the axon distal to the injury. So the injury occurs, the axon distal to the injury degenerates, it's cleared by glial cells, and in the peripheral nervous system, it's regenerated essentially. And this kind of degeneration occurs in motor neuron disease, such as amyotrophic lateral sclerosis. But while this affects the peripheral nervous system, the essentially the primary damage in this condition just overwhelms the ability for it to regenerate, essentially. And the primary damage here is also what causes the axonal damage that causes the Wallerian degeneration in the first place. Demyelination is the other type, which is the loss or destruction of myelin sheaths from axons, which impair saltatory nerve conduction. And this occurs, quite famously in multiple sclerosis, where this kind of immune-mediated attack of the myelin sheaths results in demyelination and then eventually axonal damage. Yep, so we'll do our last MCQ of the day. What cell type is primarily damaged in multiple sclerosis? So CNS neuron, oligodendrocyte, astrocyte, PNS neuron, or microglia. I'll start this interaction. Yep, and you're right with that one. It is the oligodendrocyte. So, in multiple sclerosis, immune-mediated attack of myelin, is the kind of like the primary damage, right? And it's the oligodendrocytes themselves uh, which produce myelin. So it's those. It does affect peripheral nervous system axons, neurons, but it affects the myelin sheaths first, and then the axons of those neurons. I hope that's clear. So that brings us to the end of this lecture. I finished a couple minutes early. So yeah, thank you guys so much for coming. I hope that was useful. Please, if you are able to, would you complete the feedback form? And I think Osmi will send you a copy of the lecture slides if you think they'll be useful to you. If you have any inquiries, you can email me. You can email this email address. So yeah, thank you.
Thank you so much, guys. I've left also the feedback form as a link in the chat if that's easier. Thank you. And
Thank
thank
you.
you, Jem, for your lectures. Really helpful. I joined in with the multiple choice questions. Good job. Okay.
Thank you.
Bye bye then.
Bye.